Hello, my name is Neerja Aggarwal. I am a PhD student in Laura Waller's Computational Imaging Lab at UC Berkeley. Today, I am going to present Spectral Diffuser Scope, a novel, compact hyperspectral imager we adapted for fluorescence microscopy. This work was in collaboration with my co-author Eric Markley and inspired by the proof of concept for photography developed by Kristina Monakhova and Kyrollos Yanny. In this talk, we will show you how we applied compressed sensing to snapshot spectral microscopy. We will introduce our optical setup that takes a 2D snapshot image as the measurement. We will then discuss the reconstruction algorithm we use to obtain back our full scene imaged through 64 spectral channels across a 500 nanometer total range. Finally, we will show you some simulation and experimental results. But first, let's understand hyperspectral imaging. Hyperspectral imaging aims to capture the intensity across wavelength for each point in our sample. It gives you higher resolution across wavelength than typical multi-channel fluorescence. It has many applications in fluorescence microscopy. For example, it is useful in unmixing the signal from dozens of fluorophores with overlapping spectra inside live cells, or for identifying unique spectral beads used in biological assays. There are many ways to capture this 3D spatiospectral data cube. Here we have x and y as the spatial axes and lambda along wavelength. One commonly used method is point scanning in x and y by attaching a spectrometer to a confocal microscope. Another method is scanning along wavelength by using a filter wheel with a wide field fluorescence microscope. The third method is snapshot imaging, which does not use scanning, and instead uses a spectral filter array placed directly on the image sensor. It aims to capture the full spatiospectral data cube with a single acquisition. This talk will focus on snapshot methods and adapt a technique our lab previously developed for photography. As you can imagine, these different methods of capturing a hyperspectral data cube have resolution trade-offs. Traditional snapshot imaging using a filter array allows higher temporal resolution, but usually at the cost of spatiospectral resolution. This is because the total number of pixels on a 2D camera are limited and must be split across the 3D voxels in this spatiospectral data cube. Compressed sensing allows you to recover a higher resolution spatiospectral data cube by solving for more voxels than total pixels on the camera. There are many designs for snapshot hyperspectral microscopes. These designs use a combination of filters, lenses, and prisms, requiring many additional costly components. Our system is simpler than these prior approaches and only consists of 3 additional components: a relay lens, a diffuser, and a spectral camera. These prior systems are also bulky and large, making it difficult to adapt them into existing fluorescent microscopes. Our system is more compact, only 6 inches in size, and can be coupled to the output port of any existing benchtop microscope. Now let's understand how this system works. New advances in filter technology have now allowed the fabrication of image sensors with a tiled spectral filter array. This is similar to a Bayer filter used for RGB cameras, but instead of a 2x2 grid, our filter array consists of a grid of 8x8 individual filters that make up a superpixel. Each filter is centered around a unique wavelength, and the combination of these 64 spectral filters spans the visible and near-infrared range, giving us roughly 8 nanometer spectral resolution and 500 nanometers of bandwidth. However, there is a manufacturing limit as to how small these superpixels can be. Thus, the challenge with simply placing the spectral camera in the image plane of the microscope is that we need each point in our object to land on the entire superpixel to get full spectral sampling. Otherwise, we have no way of recovering the rest of the spectra. We could try some typical approaches, like increasing the magnification or defocusing our sample to get each point to land on an entire superpixel. However, this results in field of view or resolution loss. Our approach is to use a diffuser to purposely spread and shape the light from a single point. We do this by placing the diffuser in the Fourier plane of the imaging system using a relay lens. Now, let's take a look at how we model this optical system for our reconstruction algorithm. First, the diffuser helps encode the lateral position of the points in the image plane of the microscope. Each point in the scene creates this caustic pattern onto our sensor, which then becomes our measurement. As the point moves in the scene, the caustic pattern shifts up and down. The caustic pattern is large enough that it covers multiple superpixels and allows spectral sampling. Since a linear shift of the point results in a linear shift of the caustic pattern, we can describe the diffuser's encoding as a convolution of the scene with the diffuser's point spread function. The spectral array then filters the light shining on the image sensor with a unique transmission pattern for each wavelength slice. We can represent this step as pointwise multiplication. The measurement is then a sum of each of these slices, thereby encoding the 3D hyperspectral information into a 2D measurement. From this 2D measurement, we need to recover back the underdetermined 3D hyperspectral data cube. We do this by solving a compressive sensing-based optimization problem consisting of two components, the data fidelity term shown on top that decreases measurement error, and a regularizer shown on the bottom that encourages sparsity. Given certain conditions on the scene's sparsity and the incoherence of the measurement system, compressive sensing theory provides some guarantees for successful image recovery, despite the fact that this problem is underdetermined. So in summary, we need to reconstruct the 3D spatial spectral data cube that has more voxels than the pixels in our 2D measurement. We do this by feeding our required measurement into our optimization problem along with some calibration data consisting of the diffuser's point spread function and the spectral filter array's patterns. After running the optimization algorithm for many iterations, we recover our spectral object. Here we simulated the measurement from some beads spelling out cal and tried to reconstruct the data cube. As you can see, our recovered spectra matches the ground truth. Here we show the simulation results from these cal beads reconstructed under different optical methods to get full spectral sampling, such as increasing the magnification, defocusing the sample, or using our diffuser approach. The beads are shown along X and Y and false colored to represent spectra. As you can see in the zoomed-in image of the reconstruction, using the diffuser to spread out the point spread function outperformed these other methods in terms of field of view and resolution. We built an experimental setup in our lab to test this approach. We aligned this hyperspectral imager to the side port of a Nikon TE300 epifluorescent microscope with a 4x objective and a blue LED for excitation. The imager consists of the relay lens, diffuser, and the spectral camera. Our initial experimental results look promising. Here we show 5-micron fluorescent green beads obtained from Thermo Scientific and imaged with a 4x objective. This means each point would be 20 microns in the image plane, which is smaller than an 80-micron superpixel. Left shows the measurement obtained on our spectral sensor. Note that it is hard to discern anything in this image. Middle shows the reconstruction after running our algorithm, and right shows the ground truth. As you can see, we can localize the points within a superpixel, but still obtain full spectral sampling that matches the ground truth. Next, we try and recover beads of different colors and sizes. Here, we have a scene of 10-micron green and 30-micron orange beads. We can see the 3 multicolored beads in the insets of the reconstructed image. At the bottom, we've plotted the spectra from each bead along with the ground truth. We were successfully able to recover the spectra from beads 1 and 2, however the third bead was near the edge of our field of view and likely dimmer. Therefore, it may have been more sensitive to the regularizers and will require further tuning to recover properly. In conclusion, we presented Spectral Diffuser Scope, a compact hyperspectral imager for fluorescence microscopy. We leverage compressed sensing to obtain back the full 3D spatiospectral data cube from a 2D measurement. The advantages to this approach are that it is compact, simple, and adaptable to benchtop microscope systems. It is useful in obtaining full spatiospectral sampling for a large field of view without sacrificing resolution. Some considerations to this technique are that it currently relies on bright samples, since the light from a single point is spread out and our camera is noise limited. Additionally, the scene priors require some tuning. Our future directions for this work is to develop it to image spectral beads used for multiplexing in bioassays. We also want to try using larger, customized filters for an intended application to improve throughput. Thank you.